to schedule further. Our next speaker is Susanna Gordleeva. She's a professor at Lukashevsky State University. Yeah, I'm terrible now with these on-the-spot pronunciations, so should have checked that. And the next speaker is also from there. And she'll be talking about neuromorphic computing in spiking neural-astrocyte networks. And I have no idea what that second part means, but I'm sure that you're going to explain it to us. Thank you. Mike.
Dear colleagues, good afternoon. And I am from Nizhny Novgorod, from Lobachevsky State University, after the great mathematician Lobachevsky. And first of all, I am very grateful for opportunity to give a talk on such top-level scientific meeting. And thanks very much for program and organizing committee for invitation, for Viktor Vasilievich and Max Talanov. Thank you very much. And my talk today will be dedicated to neuromorphic computing based on spiking neural networks. And first of all, I want to start my talk with for what purpose should we model in biologically relevant way the signal generation and propagation in neural networks. First of all, of course, for understanding the mechanisms and principles of information processing and storage in our alive brain. The second purpose is to build the energy efficient computing systems. And it is believed that you can read that guided by brain-like spiking computational frameworks, neuromorphic computing promises to realize artificial intelligence while reducing the energy requirement of computing platforms. And this is published in Perspective Nature and in a very recent paper. And on the slide you can see the schematic of the main principles of information, basics of information processing in a live brain. First of all, the basic primitives of this process in our brain is neurons and synapses. And we have a lot of enormous quantity of neurons in our brain, and its population account 86 milliard of cells, and each neuron is connected on average to 10,000 other neurons. And neurons can generate very short and very rare electrical signals, which propagate throughout the complex neural networks. And this provides special temporal encoding that believed to result in energy efficient computing. And on this, on the left, you can see the state of art of silicon computing system. And Viktor Vasilievich talked us about the bottleneck, which determines the physical separation of memory unit and processing unit, and on this spreading or transfer of information between this unit consume energy and time. And in brain, we have not this physical separation of the units, and information processed in one place, in physically one place, located in neurons and synapses. So on this slide, I write the key difference between artificial and biological neural networks, and there are a lot of one, and the three. One is that the artificial neural networks have generally static in structure, and biological networks are highly plastic. Artificial neural networks are typically fixed inside, while biological networks can grow and shrink in terms of neurons and synapses. And the main one is the learning algorithm, which differs from artificial neural networks and biological relevant neural networks, spiking neural networks. That's biological networks learn through experience and constantly adapt, such adapting learning, and artificial neural networks learn through algorithms that adjust weight and based on the training fixed data sets. So our brain not only consists of neurons. We have in our brain the two main populations of cells. It is neuronal cells and glial cells. The main type of glial cells in our central neural systems is astrocytes. And traditionally, it is believed that neurons are the main signal cells in our brain, and they provide the transmission and transformation of sequences of electric pulses. Astrocytes are not electrically excitable cells, but it was shown that astrocytes can display a form of cellular excitability based on variation of intracellular calcium concentration in it. And you can see on the slide the example traces of calcium activity in astrocytes and electrical activity in neurons. It is important to note that astrocytes have very large and slow signals, and the time scale of signals of calcium signals in astrocytes 3 times slowly than electrical signals in neurons. Electrical signals in neurons duration is 1 millisecond, while calcium activity in astrocyte duration could achieve seconds and 10 seconds. Model for calcium activity, mathematical model of calcium activity, is formalized in way of Hodgkin-Huxley formalism and is described by third order nonlinear differential equation systems. You can see it on slides, and main variables of it is intracellular concentration of IP3, intracellular concentration of calcium, and additional variable which describes the amount of action channels on intracellular storage of calcium ions. And this model describes calcium-induced calcium release from EPR to cytosol of astrocytes, which depends on cytosolic concentration of second messenger IP3. And on this slide summarize the autonomic calcium dynamics in astrocyte, and it is excitable element in only one different from neurons, which dynamics described by, for example, Hodgkin-Huxley or Izhikevich model. This that input-output characteristic have gradually dependence. This is amplitude of calcium signals which generate in response to some stimul, for example, for stimul of neurotransmitters such as glutamate. And this is amplitude of this stimul, for example, exciter neurotransmitter glutamate, which connected to astrocyte processes. And you can see, unlike to this dependence in neurons, we have gradual dependence of amplitude of calcium activity dependent on input intensity. With development of new experimental approach, we can see that astrocytic calcium activity in processes in soma is highly heterogeneous. And whole astrocyte could be divided in two main parts. This is soma and big processes, and little processes on distal on the soma part. And calcium activity is different from these two parts, and in main core of astrocyte we have global large calcium signals, while in peripheral processes we have very fast and local calcium activity. To investigate these mechanisms of generation calcium activity in astrocytes, we built the biophysical model of calcium activity in one compartment of the cell, and show that the frequency of generation of spontaneous generation of calcium signals is dependent on spatial to volume ratio parameter which account the morphological geometry of astrocytic process. Then we connected these compartments in the network, which connected through diffusion of calcium ions and IP3 molecules in a way that repeat the morphology of real astrocytes from real brain, and analyze the mechanisms of propagation of these calcium signals through the one astrocytes, through one morphological geometry. Then we connect this multi-compartmental model with ensembles of synaptic connection and show that astrocytes can generate the big calcium signal in its soma only in response to synchronization of activity in this synaptic connection. So we show that astrocyte can act as temporal or spatial integrator, hence detecting the level of spatial temporal coherence in the activity of neural networks which accompany these astrocytes. Then, 20 years ago, it was shown in neurobiology and neurophysiology experiments that astrocytes not only can respond to neural activity by generation of calcium signals, but when in astrocytes we can see calcium activity, it can release some chemicals, some active chemicals that named glia transmitter. And this glia transmitter is the same chemicals as neurotransmitter. It is glutamate, ATP, D-serine, and these chemicals can modulate the synaptic transmission located near this astrocyte synapse. And you can see on this picture from experiments the close location of this synapse and astrocytic process. And this finding provide new concept in neurophysiology which named tripartite synapse. And synapse consists not only from presynaptic terminal and postsynaptic terminal and also astrocytic process which can modulate the activity of synaptic connection. 12 years ago, we are one of the first mathematical group who developed the mathematical model, biophysical model of astrocytic modulation of synaptic transmission on the cellular level. And we show that different level of neural activity and different level of synaptic activity located near this astrocyte synapse can trigger calcium activity of different level. For example, if the activity of synapse not very intensive, you can see the elemental calcium response in astrocyte. When activity in synaptic connection which connected with these astrocytes elevates, we have integrated calcium response which can propagate through the cell of the astrocyte. And when you can see the synchronization activity of synaptic connection which connected to this astrocyte, the spatio-temporal integration of this calcium activity can induce the generation of global calcium response in entire astrocyte, and this entire astrocyte can in this turn modulate the activity of synaptic connections in all synaptic connections which connected to these astrocytes. And we modeled this case too, and we show in our mathematical modeling that astrocytes can induce spatial synchronization in neural circuits defined by the morphological territory of these astrocytes. Astrocytes as neurons formed the astrocyte networks, but they formed in a special way which is different from a neural network. They form this through connection by gap junction. So astrocyte networks organized as regular lattice, with connections with only local neighboring cells. So this astrocytic regulation of signal transmission in a single synapse and heterosynaptic astrocytic modulation of signal transmission and this case that neurons and astrocytes can form their levels which bidirectionally interact with each other led us to model the neural astrocyte interaction on the network level. And we model it in detail from minimal model, the example of which is present on the slide, from minimal neural astrocytic network model, which consists of pair of neurons and pair of astrocytes, and we show that astrocytic modulation can induce synchronization in pair of neurons. Then we go to the networks of large size and show the similar effect. Also, we show that astrocytes can play a role in contribution of generation of positive integrated information in neural ensembles. So what? So why brain need this astrocytic modulation of neural networks? Why does brain have astrocytic modulation of neural signaling? And what is the purpose of this modulation in terms of information processing and storage? And we start thinking about this. And a coincidence of the characteristic times of calcium dynamics and astrocytic modulation of synaptic transmission with the times of short-term memory function bring us to the hypothesis that astrocyte-induced modulation of synaptic transmission can play a role in working memory organization. And we test this hypothesis also in a mathematical model. And on this slide, you can see the scheme or cartoon of concept of our working memory in the spiking neuron network model accompanied by astrocyte. And it consists of two layers of excitable neurons which connected their synaptic connection with the excitable synaptic connection, and the astrocytic layer, and they are bidirectionally communicate with each other. Input, it is, for example, digit, is fed to neural network and induce stimule-specific synchronization in neural ensembles, which induce the generation of calcium patterns in astrocytes. After that input finish, the activity of neurons also explain, and we have only calcium patterns which exist in astrocytes sometimes, for example, some seconds. When we fed to neural networks Q, that is input that will distorted, for example, by noise, astrocyte release glia transmitters and induce the stimule-specific activation of synaptic connection in neural networks. And in this way, we can organize retrieval of stimulus in our neural network and register it by frequency of spiking activity in neural network. And on this slide, you can see architecture of a neural astrocytic network. And the one neuron is described by Izhikevich model, and calcium activity in astrocyte by systems that I showed you on the first slide. And we can teach our model to storage several information patterns in case of significant of overlapping of activity. And we present a new case of how the biologically relevant neural astrocytic network implements loading, storage, and acute retrieval of multiple items, which is important with significant overlapping. And as you can remember, I show the gradual dependence of calcium, of amplitude of calcium, of amplitude of intensity input, and this allowed us to realize the analogous memory in multi-layer neural astrocytic network, and we can leave our model to storage the original imaging in graded actin. And the system can robustly retrieve the memorized image, and even for a high noise off level. This is noise level in test, and this you can see the PCNR characteristics of our system to storage this image, for example, this gradual information. Then we integrate biologically relevant plasticity rule, spike time dependent plasticity, with astrocytic modulation of synaptic connection, and develop the system that can storage and retrieve the for example, the MNIST images. And the system, this is the scheme of the system, and it consists of two neuronal layers, pyramidal neurons and interneurons, which is inhibitory neurons. And the pyramidal neurons bidirectionally connected with astrocytic layer. And first of all, the excitatory neural networks are pre-trained with generalized patterns of all information patterns which we should load in our system, and then we turn on the astrocytic modulation of synaptic connections, and then we test our system on MNIST dataset and show that astrocytic modulation induce the 15% of outperform of the classification or correlation of the symbols in testing and in training. Which is also most important, that our system allows us to storage the overlapping information patterns. This is our team, and the first line is our young scientists, and the second line is our professors, Viktor Борисович Казанцев, Alexey Anatolyevich Zaikhin, and Alexey Nikolaevich Garbany. And I want to say by my talk only one thing, that we think that astrocytes can contribute to information processing, cognition, and cognitive impairment. Thank you for your attention.
Wow. And I have to thank Max again for yesterday's workshop, because I feel I actually understood some of this. Are there any questions? Oh, yes.
So in your modeling framework, you have a way to incorporate vasculature to complete the tripartite synapse?
Vascular what?
So these astrocytes,
Yes.
so they connect with the tripartite synapse, right?
Yes.
And that invariably includes cerebral vasculature, for example.
Yes.
Like blood vessels and stuff. So did you have a way to model this at all?
No,
Okay.
we did not compare it in the model, the vascular units.
And how exactly did you model your astrocytic layer? Like, was it some kind of a hyperparameter or an activation function, or?
Astrocyte layer is the network of diffusionally connected excitable elements, which described by systems of nonlinear differential equation, which I show on the first slides. And that is all. The astrocytes generate calcium activity and then can spread through the network. And astrocyte bidirectionally connected with neurons.
Okay.
And if astrocyte generate calcium activity, it can, for example, influence on weight of synaptic connections in this neuron which associated with it.
I see. Okay, thanks.
Right. We have
Very pragmatic question. Say, when we try to build a neuromorphic bio-inspired system, it is very clear how to make synapses.
Yes.
And even neurons. In your case, you say that these kind of cells simplify the work, modulate the work of synapses,
Yes.
correct?
Yes.
But probably it is absolutely necessary in nature, because it provides ions that are necessary to reinforce or depress synapse. But in electronic systems, do we really need it? Because we work with electrons. So we don't need to provide ions. It's just...
Yes, I understand your question. What I should say that in all our model, we did not model the effect of which you talk about. We model only additional dynamic level which have different dynamics. And only in this way we have two layers, the fast spiking neural networks and slow calcium dynamics in astrocytes. And the bidirectional interaction of these layers provide us the outperform classification rate, for example, in our system, only that. It is not about the housekeeping function of astrocytes in synapses. It's about information processing and modulation of signaling propagation in neural networks.
I think we have a question in the back.
Hi, thank you so much for the interesting talk. I'm not a neuroscientist, but I think you answered my question just now. I wanted to question that basically the way you model the astrocyte network and the spiking neural network is basically just in two layers, and the astrocyte, if I understand correctly, astrocyte network is connected directly to in some way to the neural network, and then they can influence.
Yes, exactly.
Yes,
You're right.
okay.
Yes.
I just want to make sure if I understand. Thank you.
You're welcome.
And another question.
Sorry, there's a question here. I was late for the talk, so sorry if it was already explained, but how does this compare to traditional neural networks, or the comparison in the
Artificial neural network or spiking neural networks without astrocytes?
artificial neural networks, traditional? Because you said that it compares to the MNIST classification dataset task. It says it improves. It improves your previous work on normal spike networks or on the traditional neural networks.
Yes. All our systems is not artificial neural networks at all. It's spiking neural networks, and it is the main difference. In artificial neural networks, one element, neurons, it is the analog algebraic function with binary output. In our case, neurons are excitable elements which generate rare and very short electrical signals. This is the answer. So we incorporate astrocyte and astrocytic modulation of synaptic connection only in spiking neural networks because it's biologically relevant.
Yeah, but the question was how does the results in classification of MNIST dataset compare to the traditional?
Of course, traditional or artificial neural networks outperform the spiking. Yes, of course. It's basic research. It's the future direction. Yes.
I think we had a question here in the front. Oh, sorry, not your microphone. That might be the point. Thank you.
Thank you for your talk. I have a question. In the beginning, you mentioned that there are practical applications where spiking and your neural networks can outperform traditional, like artificial neural networks. Do you have?
I never said that.
No? Okay,
spiking neural networks can outperform the artificial in some tasks.
Okay.
But for example,
of course. Do you have some example of? For me, I mean, spiking neural networks are interesting for me because I'm working on adversarial robustness of neural networks.
Yes.
As far as I know, spiking neural networks can be more robust to perturbations. Do you have some relevant results?
I very recommend these articles.
Okay.
You can
Thank you.
check.
I think we have another one in the back. There you go.
About the modeling of astrocytes, of the individual astrocytes, compared to modeling of individual neurons, I have a question. Is it also comparable in terms of there is some activation threshold?
Yes,
And
yes, they have.
what other components are there that are most important for the individual astrocyte?
It's brilliant questions, and I like it very much. And we investigate this mechanism of threshold of for generation of calcium signals, I mean global calcium signals in one astrocyte. And this threshold determines by the special temporal integration of little calcium signals which propagate from the process to the soma. This is a short answer to your question.
And in the differential equations, you have mostly some activation threshold parameter, or what are the parameters?
No, it determines by biophysical mechanisms which described by these equations.
I will see them. Thank you.
You're welcome.
Do we have any more questions? Yes. No, hold on.
Hi, thank you. If I understand correctly, using astrocytes emulates short-term memory to some extent. Did you try to employ it to actually solve some kind of simple tasks that actually require remembering some, like for example, time series texts, for example, that require short-term memory in some cases?
In our model or in animals? In our model?
Yeah, in your model.
No, actually not. Maybe we can do this. Yes, thank you. Very interesting.
Thank you.
All right. If we don't have any more questions, thank you, Susanna. It was fascinating.
Thank you very much.